Hi guys, we will know about the workflow of next generation sequencing data analysis. With modern day NGS instruments, they are capable of generating billions of reads. So billions of reads are generated in a single experiment in case of NGS instruments, and computational analysis is required to make sense of the data that seems complex. So this will break down the typical NGS data analysis workflow into three individual components, which is primary analysis, secondary analysis and tertiary analysis. Okay, to simplify the process, it is broken into three different steps. So this is the overview of NGS data analysis pipeline, which shows primary analysis with blue color and secondary analysis with orange color and tertiary analysis with green color. We will come to each of them one by one. So this primary analysis is what? This primary analysis, it involves the conversion of raw data, raw instrumentational signal data into sequence data. Raw data into sequence data, which consist of nucleotide base calls. And what are these named as FASTQ files? So in the first step, it's the formation of FASTQ files or generation of FASTQ files. So the primary analysis, it takes place on board the NGS instrument. For example, conversion of raw files, which is raw binary base call file on an Illumina sequencer to biological sequence data in the form of millions of short reads. So the first step is conversion into FASTQ files, okay, which is raw data into sequence data. And then the signal processing differs between platforms like Illumina and Torrent. So for Illumina instruments, it convert fluorescent signals into nucleotide base calls. Whereas Torrent instrument, they process signals in the form of pH change, which is converted to voltage. So Illumina instrument, it will convert fluorescent signal to nucleotide base call, and while Torrent, it will process the signal in the form of base change and convert it to voltage. Okay, so Illumina is fluorescent signal to nucleotide based calls and Torrent is pH based. Now primary analysis, it includes the pre-processing of NGS reads. Once the reads are generated, they are pre-processed, okay, after they have been converted from raw signals, and this pre-processing is done to ensure that only high quality reads of optimal length are used for downstream analysis. So pre-processing will help for quality check. So what are the steps for pre-processing? They are filtering, demultiplexing and trimming. So what is filtering about? Filtering is the reads are filtered out of the data. Okay, reads are filtered out of the data based on base call quality, which is Phred score, and the length of the read. So those which are having poor confidence base calls, they can lead to the detection of false positive variants. If they have poor confidence base calls, that is poor reads, they can lead to the detection of false positive variants. So false positive variants needs to be removed. And reads that are short, they are likely to align to multiple regions in the genome and cause poor mapping metrics. So which ones need to be filtered? The ones which are false positive variants, which have poor confidence base calls, and also the ones which are very short, they need to be removed. Next is demultiplexing. Demultiplexing refers to multiple samples being sequenced simultaneously on the same instrument. So demultiplexing, it refers to the separation of sequencing reads into separate files according to the barcode index used for each sample. So demultiplexing should be done so that multiple samples being sequenced simultaneously on the same instrument. So this is again a type of pre-processing. Okay, and the sequences are separated. The sequencing reads are separated into separate files according to the index which is provided to them, barcode index which is used for each sample. Next is trimming. So adapter sequences which are ligated to the ends of the libraries during library preparation process, adapter needs to be removed from the sequencing reads because it can interfere with mapping and assembly. Also, reads are trimmed to remove poor quality bases from the ends of the read. So poor quality bases are removed from the end, adapter are removed from the end, and this is done by Trimomatic tool, which is specially for trimming. So this was primary analysis. What is secondary analysis? So once we have found out the high quality reads, what we will do? Once we have found out the high quality reads, we will go to secondary analysis. Okay, secondary analysis is to align the reads against a reference genome or perform a de novo assembly and then call any variants detected. So many different files are used and generated during secondary analysis. Many files are generated during secondary analysis. What are these files? These files are FASTQ files. So in the first step you have seen that once the sequence reads are found from raw data, that is BCF or base calling, the file which is generated is FASTQ, which is actually generated after in secondary analysis. So FASTQ is text based file format which contains raw sequence reads and associated quality score of each base. So raw sequence plus quality score. And where are this FASTQ used? They are used for storage of raw sequence data and input into sequence alignment. Okay, for storage of the raw data and input into the sequence alignment. Next is BED, which is browser extensible data file. It is tab delimited text. That is to store genomic regions as coordinates. Okay, if we want to store the regions at the coordinates, we have to use the bed file. And in variant calling pipelines, it is used to direct the analysis to the genomic region. Next is SAM and BAM file and VCF file. SAM file is what? It is a human readable text file. It is used to store text-based information for reads aligned to a reference sequence, and it is used to store the information on read alignment. Binary alignment is what? It is not human readable because it is in binary form. It can be opened in Genome browser to view read alignment. It is used for input into variant calling pipelines. Everything will be more clearer as we move forward. Next is variant calling format, which is variant call format. It is also again a text file which stores sequence variants. And each variant occupies a single row. So where it is used, it is generated by variant calling pipelines. It is used as input into variant annotation. So I have just read it because you have to be familiar excuse me, you have to be familiar with these names and what these are. When they are used, we will do step by step. So in the secondary analysis, it's about the alignment as well. The aim of alignment is to find the genomic location where a read originates from and to determine how many reads align to that position. So there is one query which is the ones which we want to find out, and there is one reference genome, and we start aligning. For when we align, we need to know, like we want to know how many reads are aligned to that position. For example, here HG19 for humans, which is used here. In de novo assembly, no reference is used. So there are two ways of alignment. One is de novo assembly and another is with the reference genome. And in de novo assembly, there is no reference is used, and reds are aligned to each other based on their sequence similarity to create long consensus sequence known as contig. So in de novo, they are aligned to each other based on their sequence similarity to create a long consensus sequence known as contig. Okay, and again there is a problem that persists in NGS. It is that short reds can sometimes align equally to multiple locations in the genome. The longer the read, the easier it is to find the position. So short read, they can sometimes align equally well to multiple locations. The same can align to multiple locations. But if it is a little longer read, it is easier to find its position. So paired end reads, when the reds are paired end, it reduces this issue since a pair of reds has a known distance in between, which is used to validate its alignment position. If we use paired end reds, then we know what is the distance between the two paired ends, and then it is very useful to validate its alignment position to find out that if this is the particular position or not. So therefore it is very crucial to remove the reads that are very short, as I told earlier, prior to performing the alignment, as misaligned reads will lead to false positive variant calls. So this is about secondary analysis. We will continue about it in the next lecture. Thank you.